Okay, thank you. Good morning to one and all. On behalf of MSI Delhi Unit, I, Shweta Yadav, extend a very sincere warm welcome to all of you present here. I also want to extend a note of thanks to our guest of honor, Professor Dhawan Sir, Dean School of Biotechnology, JMU, for accepting our invitation and gracing the occasion. Sir has expertise in synthetic biology and its applications. We are very much excited, sir, to hear you. And surely our students will also be benefited. But before we start this event, I would like to invite Professor Deshmukh sir to say a few words.
Thank you, Shweta ma'am. Again, I will reiterate my same word. From the bottom of my heart, I'm very happy today. The reason is that Dr. Dhar is one of the well-known microbiologists in the capital of India, working in reputed university, and his topic today is also relatively new topic. So I am very happy that all microbiologists from Delhi are together here doing various activities and promoting microbiology subject, not only among the teacher, but also among the student. Now, I believe that Dr. Dhar from today is also is in our family. Microbiology Society is one family. And as Dr. Archana ma'am has told, is a very important virtue apart from microbiologist. He is a simple man, approachable to everyone. And that virtue is very useful to the common microbiologist, to the common teacher. And together with Dr. Dhar and many other scientists like Dr. Dhar, will do the much development in microbiology subject in India for the human being, for the development, for the serving the human being. And we have to create a miracle in the microbiology subject. It is possible for us. We can do it. And I'm very happy to inform everyone that many microbiology teachers and students are around me now, selflessly serving for the subject. So today, I wish all the best to the lecture will be given by Dr. Dhar. And as I've already told that this is the beginning, this is not the end. We'll do many activities in the future. Thank you, Dr. Archana ma'am and her team for arranging such a beautiful event today. Thank you. Thank you very much.
Thank you, sir. Thank you so much. Now I'll request Dr. Archana ma'am, President Delhi Unit, to say a few words. Ma'am, please.
Thank you, Dr. Shweta. I'm audible? Yeah.
Yes, ma'am.
Okay. So, very good morning to all. I'm absolutely delighted in its truest sense to receive Professor Pawan Dhar amongst us, who is the Dean's School of Biotechnology, JNU, the famous university, where a lot of good work happens. And he is a person of substance, I know personally. So we are really honored, sir, to have you amongst us. And I wish to take the opportunity to thank Professor Deshmukh, sir, also for motivating us always. Your words mean a lot to us, sir. It keeps us, they keep us going and encourage us to bring good speakers to this platform for benefit of the students. Also among students, besides students, the teachers, researchers, everyone. So I'm sure people from all over India are watching, are attending it from Google Meet or else from the YouTube, via the YouTube channel. Now, his work is, he's very famous in the areas of metagenomics and synthetic biology in India and abroad. His candidature will be read by our worthy students, so you will be getting aware, so I'm not repeating that. These fields are very great fields with the immense promise. So I urge all of you to listen carefully and imbibe the knowledge which Sir is giving by sparing his precious time for the benefit of the future generations. In the end, I would like to say that although it's a very, very tough time because of the COVID spread, but at the same time, we have to fight these things together in a safe manner, not only taking precautions for the health, but besides that, over and above that, getting involved in positive and good activities, excellent academic activities like this, because the point is that if we keep our minds occupied with good things, then also we are not getting worried or panicked from the bad news. So this is also a way to deal with the stress, at the same time gain knowledge. And we are very fortunate that through the virtual platform, it's possible to keep working, keep growing, even in a virtual manner. So not taking much more time than this, I would now like to request Mr. Abhikash Gupta, our worthy second year student, to introduce the eminent speaker to the august audience, please. Thank you. Have a great event and great day. Remain safe. Thank you.
Thank you.
Abhikash.
Thank you very much. Thank you very much, ma'am. I feel extremely elated to give the official introduction of our today's esteemed speaker, Professor Pawan Dhar. Professor Pawan Dhar is currently Dean at the School of Biotechnology, JNU, New Delhi. He also has a concurrent appointment as a professor at the Special Center for Systems Medicine at JNU, New Delhi. Professor Dhar received his PhD degree in 1993 from BHU Varanasi in human genetics. One of his significant contributions has been the development of a novel drug discovery platform from DNA sequences that were historically considered as junk. Professor Dhar serves on the Board of Referees for European Science Foundation, Executive Council Member, JNU, Biosafety Committee, Indian Oil Company, Academic Committees of CCMB, NII, and Indian Naval Academy, as well as DBT task forces of Marine Biotechnology and Genome Engineering. In the past, Professor Dhar has headed RIKEN, Japan, and TCS Hyderabad program on metagenomics, established research labs in India, Singapore, and Japan, as well as significantly published in the field of human genetics, systems biology, and synthetic biology. Sir, we all are extremely thrilled and excited to witness your talk today. Now the stage is all yours. Thank you very much.
Thank you very much, Abhikash, for this very generous introduction. At the outset, I'd like to thank Professor Deshmukh, Dr. Archana, Dr. Shweta, and Mr. Vishal, who are involved in enabling this interaction today. And of all the options you had on Sunday morning, attending a systems and synthetic biology talk is really a good accomplishment. And I really appreciate and thank all of you for being online today. So what I like to do now is I like to switch on my slides, and to make sure that I stay within the limits of my bandwidth, I will switch off the video and then I'll switch on the slides. So please give me 1 second.
I think all of us can switch off the video so that the talk quality improves.
Is my slide visible, please?
Yes, sir.
Okay, great. So once again, thank you very much for this lovely opportunity. And I hope that today, after the talk, we will have a good discussion, and we will identify areas where we can work together and grow together as a society, as well as in science and education. Before I go to the specific aspects of the work that we do, I thought it was appropriate for me to introduce the fundamental concepts on which the systems and synthetic biology is based. And also we have a lot of students here today who may have heard of these terms, but maybe they didn't get a chance to work in these areas. So please stay with me for a while when I'm introducing these terminologies and the fundamental concept as to what was the need to start systems biology, what is systems biology, and where does the synthetic biology fit in, and what kind of work is possible in future, and how much the world has progressed. So we'll talk of all of that. Reductionism has been the predominant approach of doing science. Reductionism means that when you observe an organism from the external features, and let's say this organism is moving, there is some locomotion, and there are some other features associated with the organism in terms of the color, the size, and so on. Obviously, the question that comes to your mind is to how are all of these possible? What are the fundamental mechanisms which are required to trigger these external phenotypes? So what you do is, to answer this question, you go one step deeper, and then you want to uncover the structure of the organism in order to understand the function of the organism. So uncovering the structure happens layer after layer. One layer that you are seeing is the layer of the systems. When you go a little deeper, something else comes up, something else comes up. So you're zooming in from a broader picture, you're zooming in to a narrow pixel. And the ultimate pixel that you know in this field of biology is that of the gene or the protein or RNA. Now, this moving in from the broader distance or the greater distance to a narrow point, which could be our irreducible minimum, this journey of going from the top level to the ground level is called reductionism. So when you look at the systems and you say that, okay, it's made of some tubes, some muscles, soft tissue, bone, cartilage, and so on. Now let me go a little deeper to understand how they are organized. So when you go a little deeper, what you come across is a warehouse of the organs, which are stitched together in a certain geometric fashion. And these organs coming together make a system. So when you look at the system, and then the next layer is the layer of the organs. And when you look at the organs, you want to explain how the organs are structured. You cannot explain each and everything by just looking at the organ. You got to go one step deeper. So moving from there downwards, you come across the cell. So now here is the layer of the histology and the cell biology. You move from histology, it's a low resolution, and go into the cell biology. And then you say that these organs that were looking geometric and they had certain function, they are ultimately made of cells. So if I understand cells, maybe I can understand or I can explain the function of the organ. If I can if I can explain the function of an organ, maybe I can explain the function of a system, and thereby the entire the organism. So the journey of going from the top to the bottom is called reductionism. So when you come across the cell, you look at the cell in a normal stained cell, and you see there are not much details. And you think that, well, maybe there's a nucleus in the middle and there's an empty space or not so important space surrounding the nucleus. You get one perspective of this. But the moment your staining techniques and your microscopic resolution improve, then you see a different, different view altogether, which is much more intense and which has much more details than what you thought. Now, this itself is not enough, because when you go a little deeper, you come across, for example, this organelle called nucleus. And when you go a little deeper into this, you see there is a chromosome. And you like chromosome because it is transmitted from parents to offsprings, and you think that this is something important. Remember, what was your original question? To understand how the organism is moving, and now you have reached up the level of chromosomes in search of the answer to that question. And when you look at the chromosomes, you see that it's made of DNA and histones, and DNA coil around its axis 7,000 to 10,000 times to make chromosome, and it uncoils at the end of the cell division to once again go in a relaxed fashion. So DNA becomes now your next study material from chromosomes. And early on, back in the mid-50s, when the double helical structure of DNA was uncovered, people thought that it is the mega star, and it got a huge press coverage. And so at that time, it was considered as the director of nuclear affairs, that it is the molecule that is, the most important element in the cell. So hundreds of publications are written on this. But later on, when the RNA community and the protein community caught up, and they said, no, DNA is just one molecule. We have to go through RNA. Who will deliver the information to the other parts of the cell? DNA doesn't do that. It just holds the information. But you need messengers to move to different parts of the cells. You also need proteins. In fact, the bulk of the job is done by proteins. So you cannot ignore proteins. And so over time, for the last 4 or 5 decades, DNA became the database of nuclear acids. This is not to downgrade the importance of DNA. It is just to highlight the fact that so much of knowledge is generated, and we need to catch up, and we need to see what to do after this, how to go beyond, how to use this knowledge. And the fact that DNA has been now viewed as a database, this itself means that the science has progressed phenomenally to appreciate the other assets that the cell has. So when you go beyond DNA, what you come across is chemical bonding. You come across molecules. And these molecules are, they happen to be in a biological system. So the chemistry happens to be in a biological system, and you call it biochemistry. But moving beyond that, what are the chemical compounds made of? If you ask that question, then you reach the level of fundamental physics. So here, when you want to go still further, so far you could visualize things because you could stain them, you could light them up with fluorescent probes, and you could see them, you could visualize them, you could measure the activity. But when you reach the level of atom, you realize that it is 99.9% space. Now you cannot stain the space, you cannot light up the space. It is space, it is not matter, it is space. Space does not mean nothingness. It is also made of something, but let's not get into that. That is totally different discussion. So when you reach this level, you realize that, okay, zooming out is better than zooming in because these are my limits. And the fact that atoms talk to each other, chemistry is born. And chemistry happens to be in a biological system, you have biochemistry. And then it is biochemistry talks to elements that are transmitted from parents to offspring. So you have genetics and biochemistry working together, and so on and so forth. And finally, gross anatomy is created. So now you see that the reductionism approach is pretty strong. It gave you a lot of data. It generated a lot of new knowledge. And then when you reach the level of atom, then you said, okay, let me stop here and let me go back. The whole idea was to do what is called as reductionism. Once again, I'm coming back with the term. This reductionism has been pretty successful, and it has generated a number of components that we call as RNA, DNA, proteins, and lipids, and so on. Now, when these components were in small numbers, let's say at the level of Mendel, he visualized that the presence of these, and he called as factors. He didn't see them, but he just visualized them. Amazing invention and amazing innovative mind. He did not see them personally, but he could only see the way they are transmitted from parents to offspring. So in a way, he was doing reductionism, but then he didn't have a physical tool of doing reductionism. But now we know that there are genes and there are proteins and so on. So reductionism at the level of Mendel, he could handle this with his notebook and pen and fill up the pages and then calculate and do the ratio and come with these 3 laws. But as the data increased in intensity and the density of the data was more, it is very difficult to manually keep track of it. So now you have a human cell with, let us say, close to 20,000 genes and hundreds of thousands of proteins. How will you manually do this calculations? So you got to use computers. Now you're transferring the data to the computer, and at this time, the database itself is a model. So what you're doing now is you're doing science with two different approaches. One, reductionism, which is an experimental approach. Another, which is a computational approach, using the database, investigating the data, and trying to understand the data in order to understand how the organism is functioning. Now, the third approach started more than 10 years back, and people at MIT thought that, well, these two have been very successful. One is the reductionism, another is the modeling. You do the data capture and data analysis. But the third approach, which, because they're engineers, they said, can we design the cell? Can we construct a chromosome? Can we construct DNA? Can we construct mitochondria, Golgi bodies, all kinds of organelles? Or can we boot up a cell from scratch by chemically synthesizing a cell? Can we do that? And if yes, what would be the best case scenario and boundary conditions? So this question came up, and they gave some initial proof of the concept, and then it went on very well. So the first approach we popularly know as molecular biology and biochemistry. The second approach we know popularly as bioinformatics and systems biology. And the third approach is called synthetic biology. So I just want you to get the definition accurate so that whatever I'm going to say after this, you will understand where it is coming from. We're not touching the reductionism because it's a mature science. A lot of data is there, tons of data is there. You have a high throughput experimentation nowadays, and a lot of details are available. We are also not going to spend too much time on the modeling part because that itself qualifies for at least a couple of hours of discussion. My humble attempt today is to just give you the headlines and then move to the design part. So in terms of the systems biology, this is what I played with when I joined as a faculty in Japan. This was the model that was given to us. So what you see on the screen is basically a set of equations. And these are all, this is the red blood cell is in the computer. You transfer the gene data, the protein data, the enzyme data, everything is transferred to computer, and a wrapper is created so that in a certain volume, the reactions happen. And by switching off the component, normally if you have to do this, let's say in a cell, it will take you many months and years to knock out a gene or to knock out a protein or remove a metabolite. It will take a lot of efforts. But here, all you need to do is just click the certain icon on the screen and your enzyme is off. Thereby, you can study the effect of the enzyme knockout on the other pathways. So this virtual cell that you're seeing is full of equations, ordinary differential equations. So I must also mention that when I did my PhD in human genetics from Banaras University back in '93, the bioinformatics was about to begin. It's like a tsunami coming up. So I had to train myself in a private institute in Aptech Computer Institute, where for 2 years I was learning programming and databases. In the daytime, I was teaching, clinical and human genetics to the medical students. And in the evening, I used to go and learn software development and databases. And this came quite handy when the field of bioinformatics was launched, because it needed that kind of, integrative approach. So my message to all the young people today is that please be flexible. Please keep learning. Even if it's a sort of an unrelated field, doesn't matter, nothing goes waste. Because ultimately, it's an integrative knowledge that you build up by combining individual knowledge from different platforms. So if you have to be successful, then you've got to be flexible. That is my key message. So after this, many more virtual projects came up. And one of the virtual tissue project that has made a huge wave globally is the Virtual Brain Project. And people are now trying to model all the 80 billion neurons, the billions of connectivity within the neurons, and billions of molecules within each neuron. You see, these are 3 different levels of modeling. It's a massive data capture, massive modeling, and currently we do not have a computer that can run it as a movie. So they are saying that when you reach the limits of the computing power, this whole brain may run as a slow motion movie. That much of data is there. Imagine billions of equations are embedded with billions of parameters. So it depends on your modeling method and the amount of data that you have. But it's a massive project, and the payoff is, already it has been realized to some extent. Mr. Dharmendra Moda from IBM, he used the brain data to make more intelligent chips that you can fit into robots. So when you're talking of humanizing robots in future, this is where the data is coming up. And of course, there are other methods also. But this is one of the primary motivation of modeling the human brain, is to go deeper and model intelligence and make robots more intelligent or human-like. This is not to say that modeling is the panacea of all the problems, because many times you have an incomplete understanding. Now, the sphere that you see on the screen is the actual object, and the shadow that you see is the model of that object. So what we are talking is model, not the sphere, okay? So that needs to be clearly understood. Now, measuring the shadow, the length and breadth of the shadow is easy because you can place a scale and measure it. So measurement is easy, but we have lost the dimension. So in other words, whenever you do modeling, there is an information loss. How much of information loss you're willing to settle with, that's your technique, and that's your intelligence, and that's your decision. But essentially, it is possible to model, when you have key components together, it is possible to model the behavior of the system. Many times there is incorrect understanding what we thought, and when we expand the modeling network, suddenly something else happens. So one has to keep on working on this. So I also, some time back, put a thought across, is it time to construct a periodic table in biology? If yes, then what should be the element of the bioperiodic table? So there were many, thoughts around this, and so this is still an experimental work which has not yet reached the destination, because when you have a lot of data and you want to build an engineering profile to this data, and you want to construct the system, then you've got to have certain standards, certain fixed values. And essentially, at some point in time, we may have to think of this. Unfortunately, we don't have enough data. When you go above the fold or below the fold, the data, a lot of it is missing. So it's still an island. Just a conceptual paper I want to share with you. So now the question is whether biologists can think like engineers. Why are we talking in the same breath is because there are many similarities between biology and engineering, as you can see it on the screen. However, there are many dissimilarities also. When somebody makes a motherboard for the computer, these are fixed components, right? You decide where I'm going to place the component, and how I'm going to connect the component. However, if you do the same, making a gene wire or a genetic wire, then these are mobile components. They are floating in the cell. They're not anchored to a certain surface. Like that, engineering systems are predominantly digital, and biological systems are predominantly analog. What I mean by analog and digital is when you switch on the light, this is a digital behavior. But when you are switching on the fan and then changing the speed of the fan from 0 to 5, this is analog behavior. Biology is analog, it's not digital. So one has to understand how to design an analog system than a digital system. All right, so the other things. Why it is challenging is because biological systems are noisy and unpredictable. When I'm saying noisy, I'm not talking about complaining. I'm saying there is a fluctuation of gene expression. It goes up and down rapidly. We do not have any control on that. And it's unpredictable. When you remove a gene from the cell or you add a gene to the cell, you cannot predict what's going to happen. This is highlighted by this picture that when you design an electronic circuit, you exactly know how the information is going to flow from one node to another node. You put the gates between logic gates and so on. But when you design a biological circuit, you have to wait till mRNA is made, and you have to be lucky that this mRNA, a lot of it is not degraded, so it survives for the translation event. And when the protein is made, you also have to be lucky that the protein is not channeled into different spaces and it is not degraded. It's available for the next event, which is the switching on of the next gene as a transcription factor. So now when you're designing an electronic circuit, you know it's far easier than designing a biological circuit. However, there are enormous payoffs. So that's why we like this challenge. So to design well-behaved systems in biology, one needs standards and rules of composition. Of course, this applies to both engineering and biology, but in engineering field, these standards and rules of composition have been pretty much worked out. So you have IEEE inventory or a database which will refer, and you have all the standards included there. So all the engineers, they go to this inventory and they look at the standards and they follow the standards. For example, this railway track, it has been made with this a fixed value which is existing between the rails. You cannot change the value. Once you fix it, and that's called standard. Whether it is a best value or it could have been better or it could have been different, maybe there were different reasons why the railway tracks were made within this space. Maybe there was not much space when they were cutting the trees. Maybe the engineer at that time thought that this is my reasonable space within which I can move the rail, and then it became a default standard because the locomotives were designed on that. We do not know the truth, but once something is fixed, it's fixed, and this is called standard. Rules of composition are very, very important. In what way do you combine the parts to make the entire system work? Now imagine in this watch, if your parts are not appropriately placed, the watch will not be functional. So not only the standards you also need to place them in an appropriate order, and these are called rules of compositions. IEEE standards do not exist in biology because we never thought of creating something in biology. We always thought of studying biology, not creating biology. So as a reductionist approach, it was working fine for us because you observe a pea plant, you document something, you observe a microbe, you document something, you work with E. coli, you document something. It was not creating E. coli, it was studying E. coli. That's why the standards didn't exist. There was no need to build standards. But because now there's a need, we are thinking like engineers, so we have to think differently. So it's good to use engineering philosophy instead of copying and pasting equations from the engineering textbooks simply because such solutions do not exist. Because probably engineers never faced, this is not an sort of, we're not trying to, devalue the contribution of engineers. They've done phenomenally well, flying aeroplanes and creating huge buildings and bridges and rocket science and so on. But say, for example, if you compare this with the Boeing 747 aeroplane, you will see that there are hundreds of kilometers of wire. There are 7 million parts that go into this, making the aeroplane. So it's a phenomenal complex system. But when we talk of complexity in biology, in even a single organism like E. coli, then you see that it is a far more different level of complexity because you have probability. So it's not certainty. Certainty as in electronic circuits. It's a probability. The protein may or may not reach the site. The protein may or may not get degraded. We do not know that. And there are gradient-based solutions that come up. Let's say during the developmental phase, you see the gradients are determining the formation of organs. And there is emergent phenomena, which means that individually components behave in a certain way. When they're together, you cannot predict the behavior. So this happens with biological systems. Collectively, the behavior is different. Individually, they're different. So when you have such a system, designing this is far more complex. So what's the solution? Well, it's a practical way out. In the traditional setting, we have been good in decomposing the system one part at a time, which means that you do gene knockout, you do gene knockdowns. But in the new approach, the whole idea is to compose the system one part at a time. So when you're building a microbe, for example, but it has to be controlled construction. Imagine you build, you thought you will be building E. coli, and then you end up building a pathogenic level 4 organism. So one has to be very, very clear and careful about what you're building. The concepts like truth tables have also made their appearance in the community. So when the first synthetic biology meeting was held in 2004, June 2004 in MIT, the concept of a truth table came up there, and it was proposed that we can probably go for, because E. coli has 4,000 genes approximately, so we may be able to construct 4,000 truth tables and then design E. coli from scratch. Well, conceptually, it was a great idea. It didn't work like that. People thought that by making analog behavior, by converting analog behavior to digital behavior, which means that instead of the expression going rapidly up and down and fluctuating, you give a step function wherein there is a ground level expression and suddenly it shoots up and stabilizes at the another level. So like a step. They tried to do this, it didn't work. 2 PhD students, at least I'm aware of, their work from MIT, they worked for 5 years. Well, they did produce certain digital behaviors, but then it was not robust enough to withstand the fluctuations. So people have sort of understood that designing biological systems is not going to be that easy. So one has to find some more clever approaches. Synthetic biology in its definition is a rational design and controlled construction of biological components. When I'm saying components, I'm talking about the parts, I'm talking about the modules, and I'm also talking about the networks of the circuits at all the levels. It took some time for the community to accept the word synthetic biology. In fact, it had a lot of turbulent acceptance, and people still do not accept it in certain labs. So they said, well, this is about engineering of the biological systems, right? So synthetic biology gives you a flavor as if it's a chemical biology. You're chemically synthesizing something in biological systems. So a new terminology was coined, biological engineering, which seems to be catching up much, much faster. But eventually, even if you read the synthetic biology papers or biological engineering papers, they mean the same thing. There are several opportunities available for students and faculty. There is annual international genetically engineered machines competition at MIT. As you see in the picture, students from all across the world, they come and they display their designs, and many teams from India have participated in this competition for the last 10 years and done phenomenally well. Our teams have won silver medal and bronze medal. And this, I would really encourage people to look at the website and see what all the initiatives all about. And this is mostly in the microbes. They are designing different things, and I'll come to that in a minute. So to support that, DBT has initiated this more than 6 years back. They initiated this. So it was proposed by us that we should start IBEC, which is an Indian biological engineering competition. And we made the format of inviting teams from across the country and then review their designs and their approaches, and then funding them, 10 lakh per team, and then enabling them to participate that. So we have selected people from private universities, from government universities, from across India, from different colleges and universities. So I would really encourage people to have a look at this website and think along this. Maybe it may not be this year, maybe next year. Recently, this another initiative has come up, a global alliance of biofoundries, which is being run by the UK group, and there are a lot of countries involved. I'm happy to say that we have just joined the Global Biofoundries Alliance, and we are committed to carry forward this mission forward in offering the knowledge sharing, the platform sharing, and so on. So I would encourage people to have a look at biofoundryindia.org and get all the details. There have been several microbiology applications emerging from synthetic biology. GeneCore company, for example, is using microbes to convert sugar into isoprene, and then this is used by Goodyear company to make tires. A lot of it is coming from microbes. And why they are doing that? Because environmental issues and the costing issues and so on. You'll be interested to know that people are using microbes as a part of concrete. So, which means that they take extremophiles. And they say that these extremophiles can survive high temperatures. And so therefore, these extremophiles, they've identified certain extremophiles from alkaline lakes. And they found that these can be mixed with the cement, and how do they get activated because they're in the spore form when they are mixed with the cement. So whenever the crack opens up, there is a bit of moisture. And this moisture is what invites the microbes to come there, settle down there, and proliferate. And then they have found that this is a self-healing cement, because of the property of the microbes. So I would really encourage you to have a look at that. People have gone way beyond. They are making bricks from microbes. So again, please have a look at the biomedicine.com. These are technologies which are already there. This is not a science fiction. It's already there. People are using fungus for that purpose. And so I would really encourage all the students to read this and see exciting applications emerging from this microbial science, which is so easy. It is not a difficult thing. It's very easy. Once you identify the right species, you proliferate and do these experiments. No need to do any genetic modification experiments. Just take the strain as it is, multiply that, and give different conditions, and see the miracle of microbes unfolding in this world. People have grown a lamp with mushrooms. This is a wonderful innovation from Ecovative Design company, and so many things. I think this is something that students should think and come up with the nice applications, and I'm sure you'll do much better. This is kind of a provocative slide. We have been thinking that we've always used E. coli or yeast or CHO cells as model organisms. The question is, why do we still depend on a microbe that is supplied to us by Western scientists? Of course, it's doing phenomenally well in terms of the, in terms of the model that we are using for our research and industry. Is it time for us to go beyond E. coli? Can't we design an Indian microbial cell model? So the question is that if you want to design an Indian microbial cell model, and it's not going to be difficult, please understand that. It is not going to be difficult to design such a model. It is just the intent and the thought process that needs to trigger in this direction. So if at all we have to build our own microbial cell model, what should be the properties of such a model? So these are the properties. It should be small in terms of genome, well characterized, rapidly dividing, sequence should be known, molecular tools should be available, non-pathogenic, and it should eat less. Where do you get this microbe that eats less? Because remember, why are we saying eating less? Because if you want to create a microbial factory out of this, then your input is less, the output is more, it is economically viable, and it can also withstand the conditions. So we have not yet given much attention on the desert genomics. I think this is one thing that needs to be really started in India. We have fantastic deserts which have got their own life supporting systems and marine microbes. There are hypersaline conditions where you have microbes, and we really need to push ourselves in these two directions and to see can we ourselves build one microbe which will be competing with E. coli, and of course not only scientifically but also economically. Academically, people have built cameras out of E. coli. What you see is on the left and right panel, this is nothing but E. coli, wherein there has been a slight modification in DNA. That is, when you shine light to this E. coli, it turns black because of the pigmentation. Okay. So what they have done is, this was done by the students. Again, as a part of the iGEM, they had initially displayed this. What they have done is they have taken a 35 mm camera film, and they, over the camera film, they, place the electrical bulb, and when the light is on, they placed E. coli Petri plate below the 35 mm camera film. So that means when the light is on, the dark patches in the camera film become light in the E. coli because there is no light that reaches there. And the light patches which through which the light moves in the film, they become dark. So you have dark patches in the camera film showing the light color, and the light patches in the camera film showing dark color in the E. coli. So now you have hello world and other and the PI and the students of the lab, you can see on the right panel, this is all imprinted on the E. coli. Very simple experiment, but very exciting. People are now moving beyond ATGC they want to construct a new set of building blocks. Whether they'll be successful or not, only time will tell. Visu, well, his pet name is Visu, is Narayan Analuru. He worked for his PhD for nearly 5 years in Johns Hopkins and designed under the guidance of Chandra. So Professor Chandra, he designed this chromosome. He was the first person to chemically synthesize a eukaryotic chromosome and make it lighter and chemically synthesize it, piece by piece, and stitch the pieces and reboot the cell with a functional chromosome. It was a phenomenal, contribution from this lab. Long DNA synthesis has taken the world by storm. Nowadays, you hear less about recombinant DNA technology, more about the long DNA synthesis technology, because here you have a power of redesigning the vector in the computer. So what would take you maybe months and months or years together, it can be done in 3 weeks. So what you do is you take the vector sequence, you place the markers and the other sequences in the computer. And when you combine this in a certain format, now what you do is you save this file as a text file of ATGC. You email the sequence to companies. In 3 to 4 weeks, you have a recombinant vector delivered on your table. What would take you 1 or 2 years can now be done in less than a month. This is the power of synthesizing DNA, and this whole area has really accelerated the innovation in synthetic biology. People have also, this is a very popular story, so I'll not spend too much of time on this, but one can do this precise genome editing and do the DNA surgery. People have also combined 2 bacteria into one. So this was a mega-cloning vector using Bacillus subtilis and cyanobacteria. This was, the 2 genomes were combined to create another genome, a new species called cyanobacillus, which was showing the properties of both. It has been taken to the extreme by Craig Venter. As you know, this big story came up some time back. We have a simple mission, a plain vanilla mission of making our own genes. So as you see, there has been a similarity between, a great similarity of the genes between human and chimp and human and mouse. What this tells you is that nature spent less time on making new parts and more time on reusing the existing parts. She did not waste her time in making brand new genes all the time. She said, give me a collection of genes, I'll make a new organism. And that's why you have more RNA coding genes, varieties, and so on. So the question is, how about adding more parts to nature inventory? So traditionally, by converting genes into junk and garbage, we have learned biology. The question that we asked was, given that there are 3 types of sequences in the genome, one is protein coding, another is RNA coding, and third is not coding at all. This is non-expressing dark matter of the genome. Can we make some functional genes from this dark matter? This also came from the observation that how did nature know that I have to place my start code on here and I have to place my stop code on there? How did she know that? Did she do a lot of combinations and something worked out? Maybe she retired certain genes and we call them as pseudogenes. And if that is true, then can we make more genes from the same stretch of DNA where the start and stop code on one place? Maybe we place them here and there and we take it as an information and do something on our own. To establish this, we have to know what kind of sequence. So we took intergenic sequences with no transcriptional history. They're absolutely dark matter. As you can see, the red colored lines on the screen, these are the ones we took. I'm not going into too much of details at the moment because I realize that I've got 10 minutes to finish my talk, and I'll quickly rush through the next slides. And the odds against this idea was many. This was first such attempt. They did not resemble any known gene and so on. People were not willing to work in the lab on this project. So when I explained this to my team back in Japan, because they wanted some safe idea which has a prior publication. So we had to hire Cho Su from Singapore. She was a tissue engineering expert and she had no knowledge of molecular biology. So we trained her in molecular biology. As a technician, she worked here. And she did phenomenal work because of her work, we could move forward. And then a lot of people joined in when they got the results. So the question was, how do you make a gene out of a non-expressing DNA? Well, that's a pretty simple question, because if you place the signals upstream and downstream, you have done it. But if you play with the genome at the genome sequence level, then it's a lot of work. A lot of failures are expected. So we said, let's pull them out. And then, what we did is we took the E. coli genome intergenic sequence, we amplified it, and we had a vector in vitro gen supplied, the PBAT topo vector, which already had all these signals: promoter, ribosome binding site, start codon, epitope. This is needed because when you're visualizing your protein, you need some kind of a tag to know that this is my protein. This is not made by the cell. This is made by me. And then the stop codon. So it had all the infrastructure already, though we had a lot of optimization issues later on. I'm not getting into that technical details, but probably it's sufficient to say that we did bioinformatics followed by experimental methods. And then the defining moment came when we got these proteins on the blot, Western blot. So we could see that in absence of the inducer, there is no expression. And in presence of inducer, there is expression. You see clear bands, thereby indicating these genes are talking. They're not silent, they're talking. mRNA is getting translated to protein, and we're able to catch it. Now the question was, how do they look like? So we did some computational predictions that were run in Singapore, and this was encouraging for us. The question again was, what is the phenotype of these transformed cells? And we looked at the shape, they all looked similar. And when we saw the growth pattern out of 6 samples that we had independently in parallel tracking, we could see that in one sample, one microbe, which is E. coli, the numbers were going down. So we said that if this is true, then can we switch off the gene and get back the growth? That's exactly what we found when we changed the medium, and this was an inducer-free medium, and the growth was restored. The functional study of the lab-made genes can be done by overexpression, and this is what we have shown. The Encyclopedia of Coding Elements project said that the bulk of the genome is made of transcriptome, that is RNA coding genes, not the protein coding. A very tiny part is proteome. So we are saying that can we convert not coding sequences into functional genes and proteins? There would be thousands and thousands of them. We cannot do so many experiments, which is why it has led to a computational work which we started from 2010, and it is still going on. Of course, the format has changed now. So we said that if we can profile every intergenic sequence, every sequence that does not express, and we imagine that if it was expressing, what kind of RNA and protein it would have produced, what kind of interaction, structure, function, and so on. So as a result of that, we computationally predicted antimicrobial peptide from the junk DNA of Drosophila, the DNA that does not do anything, that does not express itself. It is an intergenic sequence. And we found that it resembles a commercially available peptide to a great extent. So this was computational prediction, but when we actually did the experiments, we found that these predictions were true, because in our lab, we could kill Staphylococcus, E. coli, and Pseudomonas aeruginosa, and thereby we could find a new source of drug discovery. And this source of drug discovery was coming from sequences that were never expressed. They were called historically junk, but there is a gold mine behind that. So likewise, we worked on 3 different malarial targets, and this time we took 2,000 yeast sequences, and then we ran them through certain algorithms, and we got the top 3 hits and did the docking and so on. And when we experimentally tested them, we found that they are preventing malarial parasites from entering the RBC. And something like 60% of parasites were prevented. Now, remember, these are natural sequences. We have not modified them. So by editing them further, we hope to increase this percentage. We also found a novel peptide that kills leishmania. And this was very interesting. We said that tRNA has never translated itself. We know through our textbooks, tRNA cannot go through ribosomes and translate itself. So we said it never got a chance. It was always doing kind of an outreach activity for the last millions of years. It never got a chance to translate itself. So now, if we chemically synthesize a peptide from tRNA, what will happen? what will be the structure, what will be the function. So when we chemically synthesized a tRNA peptide from E. coli, it was an amazing result that, as you can see in the control experiments, Leishmania looks like this. But at 30 nanomolar range, which is very, very attractive from the pharmacological perspective, we could see that it is bloating and it is getting disrupted. The Leishmania is dead. The best part was not this. The best part was that even when you increase the concentration 20 times of the tRNA peptide, human cells were not affected. So that was the amazing thing. So now we are using tRNA as a source of antimicrobial peptides in future. Likewise, we found something related to breast cancer, Alzheimer's. It's currently going on, so we are in the process of patenting them. And where is it coming from? This is all coming from microbes. Please understand that. So we started a company called Foresight Biotech in JNU. It's incubated in JNU. My postdoc, who has also received his PhD from JNU, School of Life Sciences, he's heading the company, and we are committed to offer a new drug discovery platform to the community. And this, the unused genome from microbes is the source of the drug discovery. This was appreciated by European Science Commission when I was invited to give a talk at Brussels. Then the National Academy of Sciences and IUPAC, you see the IUPAC formally, they are the ones who invited. And also then it kept on moving, a lot of other things. Internationally, we have received a good recognition, but we need to really work hard and deliver, at the clinical level. So we have a certain molecular inventory. The best part is you own the IP of every molecule that you make. The intellectual property is yours. And so because this gene was never made by the organisms, it was never made in the nature based on the knowledge we have. So this is our own protein, our own gene. So you can name it after your own name if you want. So everything started from junk, so to say junk. So my students like to call this new area as junkomics. Well, these are the people who were behind my RIKEN group in Japan, who were behind this work, and now this was funded by Professor Sakaki, who was the president of the Human Genome Organization at one time, and the Kerala group, Singapore group, and JNU group, and this is currently being supported by Dr. Jaspreet Kaur from IIIT Delhi and Dr. Mriganku Das from Welltech. Well, it's a big challenge always. And the challenges are beautiful because they give an opportunity of discovering your potential. So we receive motivation from the small atom that says, be positive, even if you're always surrounded by negative things. So thank you very much for your kind attention. And I hope I have finished on time, and I'm happy to take your questions now. Thank you very much.
Thank you, sir. Thank you very much. It was a truly remarkable talk and highly effective. The way you explained about systems biology and synthetic biology from scratch, I'm sure it's going to go a long way, because not only it is highly intriguing and promises of wonderful applications, moreover, it appeals highly to us, students of microbiology and everyone alike. Thank you very much, sir. So now it is time for the question and answer session. I shall be reading the questions on everyone's behalf, and with your permission, we may commence.
Sure. Yeah. 1 minute. 1 minute only. 1 minute.
Yes, sir. Yes, sir.
Really, this lecture was amazing. Not only, I will say, amazing lecture, the content was amazing. And really, means I learned many things which I was not knowing. Although I have spent 40 years in microbiology, studying microbiology. The E. coli camera, the bricks from microorganisms, and what is a prospect in the biofoundry. And in the synthetic biology, sky is the limit, really, you have told. And many microbiologists from India may not be knowing this approach. So, sir, my request to you is that now I will try to arrange your lecture in different webinars and different conferences. Why I'm arranging? I'm a selfish. The reason is that I want to percolate this knowledge to the all students and all faculties in India. And really, we want to educate our society and teachers. I hope that you will cooperate in the future also.
Thank you very much, sir, for your encouraging words, and it's really motivating to talk to such a wonderful audience, and I will be happy to address anytime, whatever is possible.
Yes. Thank you.
May we start, sir?
Yeah, sure, please. Yeah.
Okay, sir. So the first question is being asked by Dr. Archana, ma'am. Ma'am, would you like to ask your question or should I continue?
Because you're not audible.
Okay. Ma'am, am I
Yeah,
audible
I will
now?
ask. Sorry, I was on mute. Sir, it was
Okay,
really
ma'am.
amazing and fascinating lecture. It was a much needed spark for the next generation, because now in this scenario, they really needed something very special and with a lot of promise in future. So thank you. It was like torch bearing for them. And really, I hope that's why we urge the students to attend it in huge numbers. And I will be providing the recording also to them after that. So, sir, my question is that although this work is so promising and provides a lot of sure shortcuts for big problems, but couldn't it be disturbing nature homeostasis if something went wrong and disturb the homeostasis? So could it not be very risky, because given the fact that whatever is released can't be retrieved 100%, sir? This is what my query is.
That's a very,
yeah.
Yes, absolutely. I totally agree, and it's a very, very important question. In fact, the same question was asked to me in Organization for Prohibition of Chemical Weapons when I gave their talk, in that meeting. Someone came up with the same query. Given that we do not know enough about the nature, we only know maybe 2 or 3% of whatever the nature's creation are there. When we benchmark ourselves against what is known, we are taking a very partial snapshot. All we are trying to say is that maybe whatever we are trying to create, maybe it's already created by nature in some other organism, because there's a purpose of having these sequences. It cannot be an accident that the sequences are there. We may have accidents sometimes, but it cannot be always. So there's a purpose. Through this effort, we are trying to find the purpose. And we are purely going in 2 directions, just to make sure that we are safe and we are complying to the nature's sustainability model. We are going in 2 directions. One is to create a new drug discovery platform, and the other one is to create a brand new enzyme platform. And both the models seem to be working. Now, if we were designing a new microbe for the release in the environment, that would be definitely a cause of concern. But we are not playing with the microbe. We are just playing with a small sequence of the microbe and generating a therapeutic application out of it. And the payoff here is supposedly good, because the fact that the microbe was never exposed to these peptides before, there is a hope that the drug resistance will take some time. So that is what we are now attempting. But we do not know, maybe most of the times we may fail, because when we reach the clinical trials, we may totally fail. We are totally aware of that. But at the same time, if we make enzymes for industry and we make certain molecules that finally succeed, I think that could be some contribution to the science. Yeah. But definitely nothing against nature.
Yeah, so that could be really worthwhile, especially the utilization of the introns as antimicrobial peptide and all. It could be actually serving humankind only. But what I was thinking of was that when you said that newer nucleotides human being is trying to make, so other than the 5 that we have, about that, sir.
Yes. Oh, yeah, that's wonderful. So this question was also asked to the group that is making non-natural nucleotides.
Yeah.
And well, they said that they are designing organisms which can only survive in the lab.
Okay.
They will not survive outside the lab. So even if they escape the lab, nature has no way of supporting them. And that's kind of, argument they're presenting, but we need to evaluate. We cannot take the statement as its face value. What if, mutations happen and finally they become more deadly than the natural variants? So,
yeah,
because
already we are threatened by the COVID and all. So, to not to have even a chance where somebody could release it.
Yes.
So, but like you said, if nature will not support it, then it's automatically safe. It will die a natural death. But what if nature supports it?
Yeah, that's an assumption. See,
yeah.
Not supporting is an assumption.
Yeah.
It needs to be backed up by the adequate proof. And I must also mention very quickly that people have come up with 6 nucleotide DNA, not 4 nucleotide DNA. They've added 2 more nucleotides, and they have found that this chromosome is working. It's functional. So, why they are doing it is for a lot of other reasons. They want to install their own apps, genetic apps into the chromosomes, and so that they can proliferate that. Well, there are scientific reasons. However, it needs a lot more data.
Thank you so much. It is really exciting and scintillating information. And I'm very sure the students are going to be happier than us also. Thank you so much. And wishing you more strength, more power, and more vision to continue guiding everybody. Thanks a lot. Abhikarsh, you can continue.
Thank you, ma'am. Thank you, sir. Dr. Srivastav, sir, wants to ask a question. Sir, please go on.
Yeah. Brilliant presentation, Professor Dhar. It was very nice listening to you again. I have just a few things to mention.
Sure.
First, that you would be very happy to know that we have introduced synthetic biology and systems biology at an undergrad level in the Delhi University for some time. And in fact, I'm teaching that course in the part of advances in microbiology. And this is very well appreciated by the students, because there are some exciting things and some new things that the students get motivated. We also talk about Venter's and Hamilton's experiments, the mycoplasma, minimal genomic, and all that. Now, my question to you, sir, is in the field of vaccinology, people earlier, we remember people making synthetic polio virus. And this was with an aim to make a good vaccine, where they removed all the virulence proteins, the virulence genes, so that they don't make those proteins. Now we have come a long way from that. We are now in the era where a synthetic RNA is doing the job what a full vaccine could have done. So this is in Pfizer and Moderna, as you see, and this is a very successful story. Though we had been trying to do that and trying to do this. And in this, though it's a complete secret, though no company would reveal exactly how they have done that. But I believe they are, I read somewhere in a authentic journal that they are tweaking these DNA with modified DNA that you're talking about, the nucleotides. That it's not the same RNA that we have, but they have introduced the modified bases. So I would just like to know about that technology, because it seems very promising and we have no adverse effects. And you have a platform where you can tweak the bases as the variant strains keep coming, as we are seeing. And this will be a problem always. So can you just throw some light on that? And
Thank you very much, sir. I think this is a wonderful addition to our knowledge, because if we use RNA as a basis of generating vaccine, in fact, we wanted to do the same thing. We had identified an immunologist in our school who would be taking care of these things. Very unfortunately, the last 1 year, we have not been able to do experiments because of COVID. But we really wanted to push our limits in that space, because that's a huge space. And I totally agree that a lot of exciting new products can be developed if you combine different elements. We do not know what they're actually doing, but yes, there is a huge scope. It needs to be really followed up. So I would say that it is not just a science fiction, but the fiction has been converted to reality. So now it's the time for scale. And if there are any funding avenues available, we'll be very delighted to participate in such a project. We had this in mind. We could not do this. But this is something that we'd be very interested in. Thank you very much.
Thank you, sir. Thank you so much. Over to Abhikash.
Thank you, sir. Sir, the next question is being asked by Yashodev. So he has read that there is a doctor in USA or UK, is not sure, who has succeeded in synthesis of a working human heart that works 80% efficiently. So the question is, how much success synthetic biology has acquired in cell and organ transplantation till now?
Well, I think this is very interesting. And also it comes with a huge history of such an effort being conducted. We keep hearing organ transplantation, and sometimes there are videos that talk about transplanting the brain. I would say, with the limited knowledge that I have in this area, well, for that matter, you don't need to do synthetic biology, because synthetic biology is programmed for certain other application at a different level, mostly at the molecular level. Here, the intention is to clone the organ. So to the extent of making an organoid, which means that you create a heart or a liver on the microscopic slide, this has been achieved. This much of achievement has been done. People are doing organoid culture. There's small organ synthesis where they can test the drugs. You don't need to kill animals. You can straight go to the organoid, and at a much micro level you can see what's happening to the tissue or to the internal biochemistry of this organ. It has been achieved, but people are also talking about 3D printing of the organs, 3D printing of the heart. These are exciting thoughts, but then still we don't see a market application, because there are a lot of challenges. It's not simply a thought process, but it is converting the cells into a form which should remain stable for a pretty long time till you have a donor, and also taking care of the immunological reactions. I remember early 90s, people used to think of, there were some articles in famous journals where people used to think of having orangutan as a pet, because they said that orangutan's organs are very similar immunologically compared to the humans. So next time there's a kidney failure, just remove the kidney and install it here. So like that, people have thought about it, but then it's more of a, I would say, wish list rather than a reality. So if it is a reality, then it'll be in the market very soon. So we'll have to wait for that.
Thank you, sir. There are no questions.
You
Yes,
were
yes,
talking
ma'am.
about the application of synthetic biology in breast cancer.
Yes, ma'am.
Can you tell a bit about it, sir?
So I should.
The applications, yeah, you showed in your PPT, yes.
Right. So basically, these molecules we found, they are very effective in triple-negative breast cancer, and they are able to reduce the drug concentration, which is currently given to the patient. We can reduce them by 200 times to achieve the same effect in vitro conditions. However, we'll have to do some more animal tests to see the claims if they are true at the other level. Yeah, money.
And so how?
Sorry.
Yeah, and so how synthetic biology can help in the treatment of diabetes?
Well, diabetes, if I understand, is mostly a lifestyle issue, and
yeah, actually.
Right. So synthetic biology could generate drug molecules, but then end of the day, this is a homeostasis of the system that needs to be restored back to this normal state. So I would say, yeah, lifestyle support, and also if there are certain, if there are certain targets where you need a certain drug, well, yes, it's possible to generate more drugs. If the target is defined, you can have more drugs. But at the same time, that may not cure the person. That may just temporarily suppress the symptoms for some time. Yeah.
Okay, sir. Thank you so much, sir.
Thank you, ma'am.
Thank you, sir. So the next question.
Okay. Archana ma'am, you want to say something? Okay.
Ma'am, there is actually one more question.
Yeah, go on with the
Yeah,
questions.
please go ahead.
Yes, ma'am.
Please go ahead.
So the question is being asked by Urvi Chauhan. It is a threefold question. The first part is, sir, since the field human molecular genetics is new in India, so could you please tell what scope it has in the coming 5 to 10 years?
Well, I would say molecular genetics has been in existence for quite some time. When you talk about molecular genetics, we are talking about using the molecular probes to light up the chromosome, as in fluorescence in situ hybridization, or doing chromosome painting. You paint the whole chromosome. It has applications in identifying genetic disorders and cancers. It has many applications. And it's already used in many conditions wherein your traditional chromosomal details are not clear. Molecular probing does definitely help.
So the next question that she asks is, where can I find more information about this, and what other kind of job it offers if an individual doesn't want to pursue anything in research?
Molecular diagnostics, they're not caught up in India in a big time. But, well, of course, you have molecular diagnostics definitely being associated with many hospitals where they do this kind of, regularly ELISA assay and fluorescence in situ hybridization assays. Yes, there is a bit of awareness and the market already exists. Overseas, yes, it is done at a very, very big scale. But I would say in future, if you're looking at the market value of this knowledge, then clinical genomics, maybe in few years, it's going to become a big wave once you have the reference data. So at that time, you will see a lot of applications emerging from, and still there are diagnostic tests on DNA going on, but then it is still lacking a good reference data. So once you have a reference data, then it will be a game changer.
All right, sir. Thank you. The next question is being asked by Ankit. He says, sir, you have given an example of cyanobacillus. What is the success rate of working with this kind of microbe? And isn't it unethical to produce such organisms?
Well, when a scientist thinks, they think in different dimensions. They think of, generating examples by which people will be motivated to go beyond. So the intention of this experiment was not to distribute the organism or spread in the nature. The intention was that one can use DNA as a Lego bricks and create something on the top of that. So think as an engineer. That was the whole message that the research group was trying to, because I happen to know the group and I was working very closely with them. So that is why I'm saying that the intention was to show that we need to go beyond reductionism and beyond integration and in the space of engineering genomes. That was the message, but it was not taken forward to the industry level because of obviously the environmental and other issues. Yeah.
Okay, sir. So the next question is from Dr. Lakshana Mahajan, ma'am. Ma'am, do you want to ask your question or should I continue?
Please go ahead, Abhikash.
Okay, ma'am. Thank you. Ma'am is asking, sir, your efforts will be helpful for humanity. She's thanking you. But do we have nature or conduct or compliance check committee to keep a check on work of scientists? Are these institutional ethic committees? Are there institutional ethic committees?
Right. It's a very, very important point. Once again, there are institutional biosafety committees, which we call as IBSC. They do all the checks. So even before we start an experiment, we have to submit the entire proposal to these committees. and explain what kind of sequence we are generating, what kind of sequence we are choosing, what kind of microbes, have we taken the ethical permission if we are using the human samples, is a fairly exhaustive approach, not about GNU, it's about every lab that exists in India. So you have to go through IBSC. Then it goes to DBT at much higher level. So there are at least 3 to 4 levels of regulatory checks. So currently it's taken care of. When you hear such things that, novel enzymes, novel genes, novel parts, as long as the environmental impact is not realized in terms of, releasing this in the environment, if it's at the lab level and it is a non-pathogenic level, as a scientific project, it is allowed. Only at the scientific project. Now, if you want to, let's say, redesign an organism, say, for example, there was, there was a question on the viruses. When you're synthesizing a complete virus, there has been a publication when somebody published the methodology of chemically synthesizing a poliovirus, an infective poliovirus. And this was alarming because such kind of information should not be in the public domain. There should be a limit, to how much we share, because of course we, in the spirit of, democratizing the data, one has to share, but then there has to be a certain regulation as to, but you can only regulate yourself, you cannot regulate people outside the country. They may have their own ways of doing things. But over here, all the regulatory checks are absolutely in place. And we cannot do an experiment that is not approved by the committee.
Thank you, sir.
Thank you, sir. Thank you so much.
So I don't see any more questions. We are done with all the questions, sir. I want to thank you for another question has popped up, sir. Can we take it?
Sure, please.
Okay, sir. The question is from Rajesh Kumar. He is asking, sir, you told that in biology, we don't create, but Anand Mohan Chakraborty made new species, Pseudomonas putida. So can't it be referred as a creation, a form of creation?
No, I did not say we did not create. All I'm saying is that when we create, it has to be within the limits of ethics and limits of safety. Of course, as scientists, we create all the time. But what are we creating? Are we creating for experimental purposes to show the proof of the concept? Or are we creating an organism that has to be thrown in the environment and we'd have no control over that? So Dr. Anand Mohan Chakraborty's efforts, in fact, if I recollect, this is somewhere in 80s, he had designed this organism and it was to control the oil spills. So he made an oil-eating organism. And this changed the patent laws of United States permanently. So it was a huge impact. These organisms were not released in the environment, but the power of the innovation was such that people had to change the laws. So I think it should be looked at that spirit. And there are regulatory checks, of course, at all the levels. But sometimes accidents do happen, which are unintentional.
Okay, sir, thank you. So now we are done with all the questions. And I want to thank you, sir, for taking up all the questions while being so patient and descriptive. Thank you very, very much. Over to you, Shweta ma'am.
Thank you again. And I know when I talk to the students, I get the most difficult questions. So the faculty members are generous, but students are not. And that's the spirit of in which we should move forward. So thank you very much for this opportunity, ma'am.
They are more inquisitive, and they should be also.
Absolutely, totally agree.
It was, I must say, before the official formal vote of thanks is given by the convener, Dr. Shweta, I want to really appreciate and say that it was the couple of hours very well spent, 1.5 hours, very well spent, very worthwhile, and I'm really, really very happy today.
Thank you, ma'am.
Professor Dhar.
Yes.
Dr.
Nice
Shweta, please,
Thank
over
you
to
so
you.
much. Yeah, thank you so much, ma'am. So I'm going to give the official vote of thanks, sir. Honorable Professor Deshmukh, sir, President MSI, respected Archana ma'am, President Delhi Unit, and our most valued participants. It's my privilege to have been asked to propose a vote of thanks on this occasion. On behalf of MSI Delhi Unit and Department of Microbiology and the entire supporting team, I extend a very hearty vote of thanks to Professor Dhawan sir for gracing the occasion and enlightening us with his valuable knowledge of synthetic biology and its applications in microbiology. We are grateful for the time and effort you took, sir, to share your knowledge and experiences with us. I also wish to express my gratitude to Professor Deshmukh sir for always encouraging us to organize such events. I need to mention my deep sense of appreciation and thanks to Dr. Archana ma'am for her enormous support and help for the successful organization of this session. Ma'am, I must say this event would have not been possible without you. I would also like to praise our students, Mr. Abhishek, Mr. Vishal, for their constant support and contributions. I would like to convey our gratitude to the participants for spending their valuable time with us and being a wonderful audience to make this session a success. Thank you so much. We look forward to meet you all in such events. Thank you.
And we thank Dr. Shweta for convening it successfully and smoothly.
Thank
Let me tell you one thing.
Sir, please, sir. The teachers from other colleges are also there, sir. Dr. Sunita, Dr. Shashi Chawla from Gargi College, Dr. Sunita from IHE, Dr. Kohinoor from Rajguru College of Applied Sciences, Dr. Kriti Tyagi. So many people, so many teachers are there to listen to you. So, yeah, yes, sir.
Let me tell you.
Yeah, yes, sir.
One professor from Nigeria, Dr. Ajay,
Rajguru.
he attended fully this lecture because he is regularly in touch with Microbiology Society. He is a life member of Microbiology Society. So he was also in